Okay, so I think we're about time people are not no more trickle in. So in this series we've been covering lots of work growing multiple lines and then lots of work really digging in deep to in vitro neural systems. And today, super happy, Kristen, that you could join us because you do a lot of both. So and as you let us know ahead of time, you're going to cover a lot of different things. You sent us four different references that you'll be touching on. Um, so really looking forward to it. And take it away. Thank you so much for being with us.
No, it's my pleasure to be here with this group. This is such a great community that you're getting up and running. And yeah, this is sort of a survey of our preprints. I've been making the joke lately that my lab is really great about getting stuff on bioRxiv, and we haven't been converting these into like wholesale publications. And it's really hard to communicate work that people don't see. And so this is just like my open house showing you all the cool things that we've been working on and to keep an eye out for it, please. So yeah, the talk is really broadly about using stem cells to explore the genetics of brain disease, um, and largely focused on psychiatric disorders. And I know a lot of this group is more focused on neurodegeneration. And I want to remind you that psychiatric disorders are incredibly and much too common. 1 in 5 people will experience a psychiatric disorder this year. Your lifetime risk is, in fact, 1 in 3. And they're also very severe. The lifetime disability from the World Health Organization puts psychiatric disorders in the top 5 of disorders and injuries and above neurodegenerative disease, which I think we all much more frequently think of as severe. The spectrum of diagnoses included in this umbrella is very diverse. So when I say psychiatric disorders, I'm including autism, schizophrenia, bipolar, eating disorders, anxiety, depression, and substance abuse. And there's two clinical realities that our lab is really driven to try to address. And the first is that the average delay between symptom onset and treatment in psychiatric disorders is 8 to 10 years. This is much too long to have symptoms of a debilitating disease without any sort of medical intervention. And second of all, over half of adults with a psychiatric disorder did not receive a treatment last year. And so, what we're really trying to do is apply stem cells towards this, future of precision medicine, where we can use genetics to both predict who will have symptoms prior to their arrival in the clinic, and B, match those individuals to the drug or treatment that they will do best on. I want to also point out that psychiatric disorders are heritable. Now, this is a classic family pedigree, one with a low incidence of psychiatric disorders and one in a high incidence psychiatric disorders. It's really tempting to say, well, this family has more genetic risk. But I do want to remind you that families share lots of things beyond their DNA. So you inherit risk factors from your parents, but your environment is also provided to you by your parents. And so families tend to share things like nutrition, exercise, environments, and exposures. And as we think about risk of psychiatric disorders, it actually is really important to consider how interactions between the genes and the environment um, impact not just our diagnosis, but the severity of the symptoms we experience. And so finally, these highly heritable disorders are extremely genetically complex. We'll start with schizophrenia because most of my talk will actually focus around that. Twin studies put estimates of the heritability of schizophrenia at about 80%. And we can explain about a third of that known heritability using genome-wide association studies and an extra few percent from exome sequencing. Across the spectrum of psychiatric disorders, you'll see 4 at the top: schizophrenia, autism, ADHD, and bipolar, all of which are over 75% heritable, and a few real outliers at the bottom, including post-traumatic stress disorder and major depression, which are the least heritable. That makes a lot of intuitive sense since they involve things like trauma, even in their clinical criteria. That being said, across all of these disorders, what we see is we can explain about a quarter of the known heritability from genome-wide association studies. Um, and I want to really illustrate how complex the genetics are, here again pointing at schizophrenia. So these green dots are the oldest variants that we have implicated in schizophrenia. They're copy number variants. They're very big, and they're relatively common, occurring in between 1 and 0.1% of patients. And so, given the large effect sizes and the relative frequency in the population, these were discovered about 10 or 15 years ago. Shifted to the left a little bit are these red dots. These are an order of magnitude less common in the population, so it took us about 10 more years to find them. In fact, these were discovered largely 2 years ago, 3 years ago now, I guess, in 2022. Altogether, they explain 1 or 2% of patients. Most of the known genetic risk is over here in these little blue dots. These are the common variants discovered through genome-wide association studies. They tend to be in non-coding regions. They confer very small increased risk, but in aggregate, they explain most of the genetic risk. And so here, diving in further, these genome-wide association studies, they yield things like this Manhattan plot. Here's one of the more recent schizophrenia genome-wide association studies. When you look at these, it's tempting to see a bunch of lines, each line being taller than this red line here, the horizontal line, that is genome-wide significance. But I do want to stress that each of these lines are actually collections of dots. So while here, the MHC on the chromosome 6 looks like a line, over here, this example on chromosome 4, you can really see how it's comprised of single nucleotide polymorphisms. And some of these lines are more dense or less dense, and that really just affects linkage disequilibrium or the extent to which that region of DNA is scrambled between mother and father or across generations when it's passed along. Now, these GWAS plots show a lot of information, but not a lot of it is translationally or clinically relevant yet. So how do we translate the variant, which is to say, in this vertical line, how do we figure out which single variant or variants are actually causing disease risk? How to identify the gene target or targets being regulated by those variants and the function in the specific cell type impacted. The next question that I'm really interested in is how do these risk variants interact in sum? Under the additive model, each of us inherits a dozen or more risk factors for schizophrenia, but patients might inherit 50 or 100 instead. And so it really says that we shouldn't be looking at these variants in isolation, but really their effect in aggregate. And finally, how does all of this underlie variable penetrance and expressivity? Two people with the exact same genetic risk can have very different diagnoses and different severities, even if impacted with the same disorder. And I think it goes without saying that if you and I both had the same genetic risk, we would all like to be as lowly impacted as possible and achieve our best clinical outcomes. Before diving into our stem cell models, I want to frame it around what we know about schizophrenia already. I'm going to do a great disservice here to about 50 years of clinical research. But the first thing that we know about schizophrenia is that the brains of patients tend to be smaller. This has been known for decades by autopsy studies where they just weigh the brain, and more recently confirmed in brain imaging studies like this one by Judy Report's group over 20 years ago now. Here she's highlighted in red the brain regions that are smaller in schizophrenia. It's important to point out that the brain regions are not smaller because the neurons are dying. Rather, they're smaller because the neurons themselves are smaller. You have reduced dendritic arborization in post-mortem pathology studies and fewer connections between the neurons, fewer synapses, again, in post-mortem. But that being said, this all reflects end-stage disease, the brains that the patients had when they passed away. There's a lot that we don't know, particularly what is the cell type of origin. I'll remind you that all of the currently FDA-approved antipsychotics for patients are dopamine receptor antagonists, but in fact, the genetic studies are increasingly implicating glutamatergic and GABAergic neurons as the cell type most enriched for risk variants. And so what's going on is disease starting in glutamatergic and GABAergic neurons and then subsequently affecting dopaminergic neurons. What other cell types are they in fact impacting and how? And why do we not know something as fundamental as what cell type goes wrong first in schizophrenia? Well, the truth is, and I think this is an obvious fact for everyone in this audience, there's not enough live human brain tissue for studies of disease risk and drug discovery. I like to point out that while mouse models have been tremendously informative, I was not trained as a neuroscientist, and even I can tell the difference between this mouse brain and a human brain, and that remains true even when we scale for size. I do think it's important to highlight the strengths of alternative models, and I think what mouse models have been great at is demonstrating causal links between gene mutations, circuits, and behavior in rodents. And every model has limitations. We all know stem cells limitations. And I think on mouse models, their limitations are that it can be really challenging to study the impact of non-coding common variants, simply because many of these common variants are human-specific and not frequently conserved between rodents and mammals, or rodents and humans. Second of all, as anybody who's tried to cross two mouse transgenes will say, it is really difficult to engineer and breed complex crosses. I'm not talking about two transgenes now, I'm talking about dozens of common risk variants in combination. And finally, how do we really assess pleiotropy and variable penetrance of complex and uniquely human phenotypes like psychosis? And so I think here are some of the limitations of mouse models and why I started moving towards human stem cell models. The basic idea here, as all of you know, is that we are taking skin cells or blood cells from as many people as possible, turning those into stem cells, and then adding genetic risk to stem cells from controls and subtracting genetic risk from stem cells from patients using CRISPR engineering, and then using those isogenic comparisons, so cells made from the same genetic background but with more or less genetic risk, and using those to make all the major cell types of the brain. The really important comparison is the cells that we're making to those in the human brain, asking how good the cells are. This was analysis that we did a long time ago, but many, many people in the field have done the same analysis at this point now. Briefly, in blue, we've taken all the RNA-seq that was available circa 2015, 2016, including repositories from GTEx, Common Mind, and the Allen Brain Span. They're colored here in blue with the fetal darker white.
Can people hear me? Did we lose Kristen or did we lose
Stem cells
me?
young. So we're modeling predisposition to brain disease using stem cell derived neurons. One really quick highlight of some very old data showing that if we make stem cells from cases and controls, at this level of magnification, there's no obvious difference between cells from cases and controls, but there are subtle effects, including reduced branching and reduced synaptic activity, all of which are quite consistent from what we know from postmortem analysis. And so the question here, why are the cells firing differently in a dish? Do the molecular causes underlying these in vitro phenotypes represent genetic risk factors for schizophrenia? Or, put another way, can we use this as a platform to decipher the genetic risk factors of schizophrenia in vitro? And so that's really where the lab has been for the last 10 years or so. The first postdoc into this space, Nadine Shroud, I will forever look upon as the bravest postdoc in the lab because she took the PGC2 analysis from circa 2016 and picked a single SNP to validate in vitro. Now, how did she pick from amongst the hundreds of SNPs over genome-wide significant? Well, she worked with a bunch of really great geneticists, and we asked for two criteria out of each SNP in one of these vertical lines. We asked across all the SNPs, which one was most significantly associated with disease risk. So in that case, it would be, by definition, the top SNP in a column, but also which SNP was associated with changes in gene expression of a nearby gene in the postmortem brain, under the assumption that these non-coding SNPs are regulating expression of a nearby gene. And in fact, across the entire PGC2 with 145 of these vertical loci, only one loci showed this effect of a single SNP in the top right-hand corner that was the most significantly associated with disease, but also with expression of a nearby gene, in this case, furin, in the postmortem brain. This SNP is called RS4702. And to highlight how unique it was that we found a loci with a single putative causal SNP, I want to show you the number two loci in the PGC2. The number two loci occurred near this gene SNAP91. And here we've got, 2 or 3 dozen SNPs localized in the top right-hand corner that were all together most associated with disease risk and brain expression of SNAP91. And so it's really hard in this instance to find a single SNP worth editing. And of course, given the distance between all these variants, we couldn't edit them in combination. And so Nadine's going to use CRISPR editing to target RS4702, and we'll come back to SNAP91 later in the talk using CRISPR activation to push it in the disease-relevant direction. So looking more at RS4702, it took Nadine a couple of years, but she achieved perfect edits of this non-coding SNP in 2 control donors. GG is the disease-associated genotype and was predicted to have less expression. It did, glutamatergic neurons. Over the 2 years it took Nadine to edit this SNP, it was discovered that the SNP lies in a microRNA binding site in the 3' UTR of furin. Here we've inhibited that microRNA and obliterated the SNP effect. And so basically what we're showing here is actually a context-specific EQTL, one where there's a differential regulatory activity by genotype only in the presence of miR338. Finally, these GG neurons have reduced dendrite outgrowth and reduced firing, consistent with the postmortem and the previous IPS cells I already highlighted. That, however, took us about 3 years to do. And over the course of those 3 years, the PGC2 went from 145 loci to the PGC3 with 250. And we didn't scale our ability to test variants. And a lot changed in the lab over that period. I started being more interested in not necessarily in testing just these linear genotype-phenotype relationships, but trying to understand what bends these curves. What are the environments in which phenotypes associated with a genotype are made worse? And what are the environments reciprocally where these phenotypes are made less severe or smaller? And can we use this to bend clinical outcomes? And so the lab over the last 5 years has really been focused on this question of how does context modify genetic risk? And we're going to use context really broadly. So one instance of context will be donor genetics. So how do all the other risk variants in your genome impact the phenotypic effect of any single one of them? Second of all, how do risk variants have effects across neurodevelopment? Do they act during field development, childhood, adolescence, or in aging? How do risk variants have different effects between the cell types of the brain, whether that be in microglia, astrocytes, or neurons? This is a really interesting and ongoing question. How do risk variants have different effects by sex? We know many psychiatric disorders, such as depression and Alzheimer's, are more common in females, and autism and ADHD more common in males. And finally, the largest group in the lab is really interested in gene by environment effects. So how do things like stress, inflammation, injury, metabolism, drugs, and toxins impact genotype-phenotype relationships? And so the first story that I'm going to talk to you about is really about stress and inflammatory effects and as how they associate to brain disorders. So we know that stress increases risk for psychiatric disorders, but stress alone is not causal. Unfortunately, about 1 in 3 of us will experience a trauma during our lifetime of sufficient magnitude to cause or to be associated with PTSD. But not all of us will subsequently be diagnosed with PTSD after trauma. In fact, only about 10 or 20% of people who experience a trauma will go on to develop PTSD. We don't truly understand why some people are more resilient and some are more susceptible following trauma exposure. It's clear that some of this relates to prior exposure to trauma, particularly childhood traumas, which will lead an individual to be more susceptible. But one of the open questions we wanted to ask is, how do our genetics determine how we respond to traumatic stress? And so this was a study started a long time ago, really foundationally led by Rachel Yehuda at the Bronx VA, and with a critical partner being the New York Stem Cell Foundation, and they made all the stem cells. The bioinformatics analysis I'll talk about was led by a former MD PhD student, Karina Siao, in the lab, who was co-mentored by Laura Huckins. So here we have stem cells from 39 donors that Rachel recruited. They were all combat exposed, in this case, male veterans at the Bronx VA. NICEF made stem cells from all 39 donors, and then we treated them with vehicle or with low or high dose synthetic cortisol. And the first thing that I think you can see is that the more we stress neurons, the more they change gene expression. That being said, half of these neurons came from individuals who had PTSD, and half came from combat exposed controls who did not have PTSD. And when we break down this very same data now by diagnosis, we see the opposite trend, which is gene expression differences by diagnosis were greater at low dose and less at high dose, which I think is really saying everybody's neurons respond to high dose stress. But the neurons from people who had PTSD were hypersensitive to stress at low doses. Um, in fact, those genes that are changing at low dose stress, particularly in the neurons from PTSD cases, were enriched for a number of PTSD gene sets, including those from postmortem and genetic studies. They weren't specific to PTSD. In fact, we've got also highlighting genes linked to developmental disorders, schizophrenia, epilepsy, and autism. And so overall, we think that stress is dysregulating psychiatric risk gene expression in neurons. But how? Does stress then dysregulate the risk loci gene expression in neurons? And again, I'm going to come back to this idea of base EQTL. So I'm going to explain it a bit more. I mentioned already in the context of furin, at the furin RS4702, the GG genotype was associated with less expression and the AA with higher expression. We're going to call that a base EQTL at baseline. And now we're going to introduce the idea of a stress interactive EQTL. Again, EQTLs typically are in the promoter or enhancer, although that furin example was unique, being in the 3' UTR. In the absence of stress, or at baseline, again, less expression for the GG allele, more for the AA. But in this toy example, in the following stress, the GG allele drives more expression and the AA allele drives less expression. Is this possible? Does this sort of thing exist in biology? And in fact, it does. Led again by Karina and very generously supported by Matt Giganti, who provided us access to RNA-seq from 304 brains, now combat-exposed veterans at the New Haven VA. And Karina asked, were there genotypes that showed differential expression of nearby genes only in those veterans who experienced trauma and not in those veterans who did not experience trauma? And in fact, she found about 9,000 examples of this genome-wide that regulated about 900 genes. This is across brain regions. What's even more interesting is that she was able to validate half of these in stem cell-derived neurons. So now she's taken those 39 donors, we've turned them into both the glutamatergic neurons I mentioned earlier, as well as GABAergic neurons, and treated them with H-court for 24 hours. And in this data set, we replicate half of the stress responsive E genes in the postmortem brain. This is truly unexpected. And let me walk you through why I was so surprised to see this. First of all, the postmortem brain data set comes from actual humans where their neurons are part of circuits. And it was a real world combat trauma that happened years or decades before the patients passed away. In the stem cells, we have artificial neurons made in a dish, treated with the hormone for 24 hours, and then immediately harvested for RNA-seq. And they're also immature neurons, as I mentioned earlier, relative to the adult ones here. And yet half of them are replicating, which really suggests to me that stress response or trauma response is conserved across neurons independent of their developmental maturity. And so altogether, we're putting forward a list of 139 novel risk genes for PTSD that we think are only risk genes in individuals who've experienced trauma. So diving deeper mechanistically, how is trauma response genetically regulated? Well, we thought it might be mediated by transcription factors. This makes a lot of sense. Maybe stress increased the levels of a transcription factor or the activity of a transcription factor that then preferentially bound regions of the DNA that had these PTSD risk factors. That's exactly what we saw. So we see increased activity by NF-kappa B, transcription factor linked to stress and inflammation, and likewise the glucocorticoid receptor, which again makes sense since we were putting its ligand, cortisol, onto the cells in a dish. Here's a third example, YY1, another well-known stress responsive transcription factor. And to validate the effects of YY1, here's RNA-seq from 2 controls just treated with H-court. And so you can see genome-wide, there's some genes that are up and some genes that are down when you treat control neurons with our stress hormone. And here is the matched RNA-seq from the same donors when we add the same stressor in the presence of a short hairpin for a YY1. And you're really just blunting the effect. Neurons are not responding to stress as strongly when we knock down YY1. So again, we think these transcription factors are mediating stress responsiveness in neurons. At this point, Karina graduated. And I'll tell you about some follow-up work led by another former PhD student, Kayla Townsley, also co-mentored with Laura Huckins. So whereas Karina was really interested in trauma that impacts adults, Kayla was really interested in maternal immune activation and how the environment that the mother experiences while pregnant will go on to impact the clinical outcomes of the fetus in childhood and adolescence. And so we have known epidemiologically for decades that women who are pregnant during famines and war and pandemics are more likely to give birth to children that will be subsequently diagnosed for autism or schizophrenia. But the mechanisms for this are largely unclear. Yeah, in the mother, these changes are associated with increased cytokines and stress hormones. So here we have IL-6, interferon alpha, and H-court. We're treating neurons from just two controls with these three different stressors. And I want to point out that the stress responses are unique across these three contexts. And it's not just the magnitude of the response, there's different genes that are responding under each condition. And so it's not just that we're seeing neurons that at baseline and reactive, we're seeing differential responses to these differential cues. And so the question that Kayla spent her PhD working on is, do our genetics determine how we respond to prenatal stress? And to do this, rather than recruit a large cohort of kids who were born from mothers who had experienced different stressors and traumas, we did this in vitro using a massively parallel reporter assay. This is an assay that asks what is the regulatory activity of a non-coding region, such as those identified from GWAS. And so Kayla designed a library of about 7,000 non-coding regions from the GWAS of schizophrenia, anxiety, depression, PTSD, and Alzheimer's. In this library, so a classical non-coding screen would insert the non-coding region behind a luciferase and ask if it increased or decreased the extent to which cells glowed. It's not very scalable. And so here, rather than using luciferase as a readout, we're going to use RNA to DNA ratios because each variant is barcoded in the library. If there's more RNA than input DNA, we're going to assume that non-coding region is in a promoter or an enhancer. And if there's less RNA than DNA, we're going to assume it's in a repressor. Typically, these MPRAs are done in cancer cell lines, but Kayla's going to attempt one of the early MPRAs in neurons. Not only that, she's not going to do it at baseline. They're going to do it under dynamic cues across IL-6, interferon, and H-court treatments. So the first thing that Kayla found, again, is that H-court is broadly dysregulating psychiatric risk loci activity and some specific responses to interferon and IL-6. We can dig much more deeply into this at the level of individual loci. So here we're going to dive into one loci on chromosome 2 near the contactin 4 gene. So at this loci, there's actually a number of disease-associated SNPs, including SNPs for schizophrenia, Alzheimer's, and depression. At the particular hit that Kayla found at this loci, you can see that the C allele is in the middle of 3 different binding sites, one for CREB, CREM, and REST. I'll note they're all repressors. And the T allele is in a transcription factor, a motif for ATF3. And so, and finally, I'll point out that at this top hit loci, you have a H-court specific responsiveness, and we don't see strong responsiveness for IL-6 or interferon alpha. And so overall, what we're really seeing is that that C allele gets repressed more strongly under stress conditions, whereas that T allele gets activated more strongly under stress conditions. And so we really have this like bimodal genotype dependent stress response at this particular loci. And so overall, what I think I've shown you so far in this talk is that it's not just about sunny and stormy weather, but that the type of weather matters. Whether it's blizzarding, windy, or lightning out will impact your risk for different diseases differently. The next kind of vignette in this story goes from gene environment interactions to gene gene interactions. It was again started by that former postdoc, Nadine Shroud. I had mentioned that furin was worth editing, but the next three genes we couldn't edit the genotype for. Instead, we just perturbed them using CRISPR-A in the disease-associated direction. And so what Nadine did was a single gene RNA-seq for all four of these genes, and then summed the RNA-seq profile to get an expected additive model of what she thought would happen if we were to perturb all four genes in combination, the way they are inherited in a polygenic risk model. And then in parallel, she actually conducted the combinatorial perturbation in the same batch. And she asked to what extent that model predicted the four gene effect. It turned out the model was pretty good. About 80% of the time, genome wide, the gene expression changes matched the expected additive model. About 20% of the time, the changes did not. 7% of genes were more down or less up than expected. They were enriched for the major neurotransmitter classes. Likewise, 11% of genes were more up or less down than expected, and they were enriched for the rare and common variants associated with schizophrenia and bipolar. And so overall, what I'm saying is if you want to understand combinatorial effects and you're just testing genes one at a time, you're going to get about 80% of the relevant biology per dollar spent. But the data that you're missing will be enriched for the very disease that you're studying. The one major caveat to this study was that we ranked the genes entirely on the strength of statistical evidence, which is to say we didn't really pay attention to the fact that 3 of the 4 genes have roles in synaptic function and how that might have confounded the observations. So after Nadine started her own position, Michael Deans took over this project. In the interim 5 years, we'd progressed from PGC3, from PGC2 to PGC3, and to a much larger reference EQTL panel. So instead of having 4 genes to prioritize, Michael had his pick of 25 genes. From those 25 genes, he picked the top 5 synaptic genes, the top 5 epigenetic or regulatory genes, and this is, of course, because the major risk factor, the major classes of genes linked to schizophrenia are synaptic and epigenetic. And then finally, he picked 5 multifunction genes. These 5 genes were not synaptic, not regulatory, and had no apparent relationship to each other. And then he perturbed each of them alone and in combinations of 5. The synaptic genes behaved exactly as Nadine had observed, about 80% additive effects and 20% non-additive. The regulatory genes were likewise at 80% additive. The only real surprise came from the multifunction set, which performed exactly in an additive model. So I'm going to walk you again through conceptually how we're thinking about this. This is an example, a toy example of 10 risk genes with individual RNA-seq and an expected additive model. Overall, we're increasingly convinced that we see a measured combinatorial that adds up to just less than the expected model. Towards explaining what genes were not responding as much as expected, we asked about transcription factors, expression levels, pathways, and the only real connection we found, the only enrichment we observed, was in a class of genes we're calling convergent. Now, if these non-additive genes came by comparing single and combinatorial perturbations, the convergent genes are going to derive just from looking at the single gene perturbations. If we ask across these 10 genes, which differentially expressed genes were up 10 out of 10 times or down 10 out of 10 times, so downstream genes that always co-occurred across all 10 genes, we're going to call those black dots, in this case, convergent genes. And these convergent genes actually are over-enriched in our non-additive genes. And so it looks like this. In our synaptic genes, we have a huge number of our convergent genes overlapping, ditto in our regulatory genes, and very few convergent genes that didn't overlap with our non-existent, non-additive genes in the multifunction. So now we have a list of shared downstream targets across diverse schizophrenia risk genes. You know, translationally, it's hard to imagine targeting every risk variant that a patient might inherit, especially if they're inheriting dozens of them. But if dozens of their risk variants are converging on a smaller number of gene targets, perhaps we can target those convergent genes to reverse schizophrenia signatures. And so that's what Kayla did for us. She took our convergent networks and then looked in the links data set made available by the at the Broad and predicted gene or drugs that would reverse the expression changes in our convergent targets, not our schizophrenia risk genes, but those downstream shared convergent targets. One of their top hits was atoxamir. And so Michael put this on the cells. Here's an example of the Callan CRISPR A, so disease direction Callan N1 perturbation. And with atoxamir, you're blunting and even reversing that transcriptional effect. Likewise, with the second schizophrenia gene TMEM219, in green is the RNA-seq of just TMEM219 activation. And here it is together with atoxamir, a blunted or reversed effect. And so we think there's a possibility here that we might be able to target convergent genes downstream of shared schizophrenia risk genes. The next question that the lab had was how convergence changes between cell types. We're going to pivot a little bit here. And instead of focusing on GWAS target genes of small effect, we're going to look at loss of function mutations that are not inherited in combination. So loss of function mutations inherited in autism, and ask if they likewise converge. So now we're going to use a pooled CRISPR library of 29 genes, all of them autism-associated genes, and most of them chromatin modifiers, and ask if there's convergent genes downstream of them. In fact, we do see convergence. Mei Lin tested this list in neural progenitor cells, immature eye glutes, mature eye glutes, and GABAergic neurons. And the first thing that you can see is that the effect of these ASD gene knockouts cluster by cell type. So knocking out the same autism gene in 4 different cell types is less similar than knocking out 4 different autism genes in 1 cell type. The extent of convergence was strongest in mature glutamatergic neurons. It's really interesting because other groups have published previously looking at convergence in NPCs in autism genes. And we see that, but it's much stronger in glutamatergic neurons. What types of genes are convergent in glutamatergic neurons? Of course, unsurprisingly, there's a lot of synaptic hits. The interesting thing to note here is that we didn't over-enrich for synaptic genes in our gene list. In fact, we're over-enriched for chromatin modifiers. So these chromatin modifiers are converging on synaptic genes. Of course, they're also converging on epigenetic genes. And there's some other functions, such as mitochondrial function and signaling that we also see coming up. We really wanted to know if this molecular convergence that we're seeing in vitro with human neurons predicted anything about behavior in vivo. And so here we're working with Ellen Hoffman. What was great about Ellen's work is that she works in zebrafish, where it's really scalable to knock out a lot of autism genes. In fact, of our list of 29 autism genes, she's already knocked out over half of them. And she's got behavioral data on again, over half of our autism genes in the library. And so April Pruitt, a grad student co-mentored by Ellen and I, has clustered the zebrafish mutations based on shared behavioral characteristics in the water when you knock out autism genes. And so you can see that we've got 4 real clusters of autism genes showing up. And then Kayla reran our molecular convergence based on the human neuron RNA-seq of these autism gene perturbations within each cluster. And so you can see that the cluster 1 genes, Norexin, MBD5, and KDM5B, have convergence that's largest in glutamatergic neurons, whereas cluster 2 shows convergence that's perhaps greater in GABAergic neurons. Kayla again predicted drugs that might reverse the different convergent effects in each cluster, finding, for example, that they predict perfivazine to reverse glutamatergic neurons in SET2, or naltrexone to reverse glutamatergic neurons in SET2. And then we actually tested the effect, or we looked in Ellen's existing data set of the effect of these drugs on behavior, since this was all part of Ellen's existing data sets. And you can see the extent to which some of Kayla's predictions actually had reversal effects in the behavior of zebrafish. And so now this next section, I'll close by pointing out that we have different types of stormy weather, but it might also be possible to package sunshine and rainbows in a pale form to minimize phenotypic effects. And the last idea I want to close with is, what about when mutations have divergent impacts? I've been talking so far about GWAS hits perturbed with perturbed changes in direction of expression or loss of function mutations in autism. And so we'll narrow down to one specific autism-associated gene, Nerexin-1. Nerexin-1 is actually highly associated with autism, but also schizophrenia, Tourette's, bipolar, epilepsy, a number of psychiatric disorders. So the effects of these heterozygous Nerexin-1 deletions are pleiotropic and variably penetrant. Nerexin-1 is highly alternatively spliced with hundreds of isoforms known in the mouse brain even 10 years ago, with the greatest splicing complexity in brain regions with the greatest cellular complexity. And more recently, Thomas Sudov's lab's done a really great job demonstrating that the heterozygous effects of Nerexin-1 are human specific. So most of the behaviors with Nerexin-1 knockouts in mice were all found in homozygous nulls. And so in this particular example, Thomas Sudov's team engineered the same mutation in Nerexin-1 in mouse and human stem cells, turned those into fibroblasts, reprogrammed those back into stem cells, made eye glutes in both mouse and human cells with the same mutation, and then patch clamped those. And what you can see is in identical eye glute neurons with the identical mutation, you have an electrophysiological phenotype in human neurons, but not in mouse neurons. So again, I think this is a really interesting mutation with a human-specific effect. Aaron Flaherty, a former PhD student in the lab, started this project 10 years ago by asking if we could even see complexity in the human Nerexin-1 splice repertoire. This is long-range sequencing of the Nerexin-1 repertoire. We've ranked the isoforms from most abundant to least, and we've colored in orange those isoforms that were expressed both in the human brain and in stem cell-derived neurons. And so what I think you can see is that the most abundantly expressed isoforms tend to be shared between the human brain and stem cell-derived neurons. So Aaron went forward with this using a stem cell cohort. The last thing I didn't mention about Nerexin-1 deletions is that they're non-recurrent. Unlike 16p or 22q11 that tend to have the same boundaries every time we detect them clinically, each Nerexin-1 carrier has a deletion in a different part of the gene. Here we've got biological replicates only because they were inherited in families. So this is a mother and her son with 5' deletions in the promoter and first 2 exons, affected by bipolar with psychosis in both cases. And here's a monozygotic twin pair with deletions from the 2nd, 3rd, and 4th from last exons, both diagnosed in childhood or adolescence with schizophrenia. So we've got biological replicates of 2 very distinct heterozygous deletions. Aaron made stem cells from all of them, could find pretty robust decreased neurite outgrowth in the patient neurons and reduced activity in neurons, again, from the cases, whether they be 5' or 3'. Towards dissecting the mechanism responsible, she asked to what extent these phenotypes reflect perturbations in few or many Nerexin-1 isoforms. Put another way, are these patient-derived neurons firing less because one really critical Nerexin-1 isoform is down in the hets, because all Nerexin-1 isoforms are down in the hets, or something else going on altogether? And so back to long-range sequencing, here in orange, we're seeing that about half of the wild-type isoforms were significantly down in patient neurons compared to controls. Another third were not even detected in patient neurons compared to controls, which I think is a sequencing depth thing. So overall, mass dysregulation of the wild-type Nerexin-1 isoforms in our 3' cases. But something really unusual, the most abundantly expressed isoforms in our 3' cases were mutant isoforms caused by splicing across the deletion. And so we wanted to ask if these mutant isoforms were functional. And so Aaron cloned them and overexpressed them in lentivirus and control neurons, and was able to show that overexpression of a single mutant isoform was sufficient to impair activity in control neurons. In our 5' deletion carriers, who we don't believe make mutant isoforms, overexpression of a single wild-type isoform was sufficient to rescue. But in those 3' carriers who are impacted by both a heterozygous deletion and mutant isoforms, we couldn't make them better by overexpressing wild-type isoforms, and we couldn't make them worse by overexpressing mutant isoforms. And so we ended this story really concluding that we thought phenotypes were occurring through 2 independent mechanisms: a loss of Nerexin-1 dose in all cases, and the additive effect of mutant isoforms in a subset of cases. Now, what were the odds that I found the only 2 Nerexin-1 cases with with gain of function mutations? I don't think I'm that lucky. We looked into the PGC2 dataset at the time, and our best estimate, just based on those cases that impacted Neurexin 1 isoforms that we thought would make mutant isoforms that were translated, we think about 20% of cases might have potential mutant isoforms. Aaron at this point graduated, and Michael Fernando, another fantastic grad student, co-mentored with Paul Schlesinger, took over and really pushed the story in a more translational direction. So same stem cell lines. First of all, Michael repeated the key finding from Aaron's study, now specifically in pure glutamatergic neurons, reduced activity by MEA. Uniquely or newly found in GABAergic neurons, he found the opposite phenotype, hyperactivity in immature GABAergic neurons, again by multi-electrode array. Michael trained as a patch clamp electrophysiologist, and he reproduced these findings by patch clamp. So reduced EPSC frequency in glutamatergic neurons from cases, and hyper iPSC frequency in GABAergic neurons from cases. He validated these through our best possible isogenic designs. Now, this mutation is too big to correct by CRISPR, but you can knock down control isoforms by short hairpin, and that allows you to recapitulate reduced EPSCs and increased iPSCs. Likewise, you can knock down mutant isoforms, trying to recapitulate them to more like a control state, and that is sufficient to increase EPSC frequency in eye glutes and decrease IGABA hyperexcitability. And so to close with, Michael really wanted to try to mechanistically rescue these neurons. He went after rescuing the loss of function and gain of function aspects of these patient mutations through different approaches. To rescue loss of function, he asked if he could increase expression from the wild type allele, guided by a recent ChIP-seq experiment that showed that the estradiol receptor bound just upstream of Neurexin 1. He tried adding estradiol to our patient neurons and was able to show that you increase Neurexin 1 expression in glutamatergic neurons treated with estradiol, and that this was sufficient to increase EPSCs and glutamatergic neurons. There's a significant increase that brought it back to almost wild type levels. To target the gain of function, he designed a number of antisense oligos, all of which were sufficient to reduce Neurexin 1 expression levels in glutamatergic and GABAergic neurons, but unfortunately, they were challenging to patch, and so we don't have a full rescue here. But altogether, I want to leave you with the idea that sex hormones can modify the impact of genetic risk, and this matches with a lot of clinical data showing biases across these disorders, increased autism and Tourette's and ADHD in males, and Alzheimer's and depression in females. And so with that, this idea that I want to close with is really that gene-gene and gene-environment interactions will inform genetic resilience, allowing each of us to achieve the best clinical outcomes possible with the genetic cards that we were dealt. I want to stop and pause and thank largely Michael, Kayla, Karina, and Maylin for the data and analysis I talked about today, and my really fantastic collaborators, including Gong, Rachel, Paul, Laura, Matt, and Ellen. And with that, I'm really happy to take questions from this group. Thank you so much.
Thanks, Kristen. You gave us a ton of things to think about here. So for sure now, I'm sure we'll have some questions. So please just jump right in.
Maybe I can start the ball rolling. Kristen, that was just sensational. So I had the pleasure a few weeks ago of listening to a colleague of yours presenting this stress story. It's really amazing, beautiful. The question I have for you is related to a guy in Canada called Michael Meaney, who was saying, decades ago, that the hippocampus played a critical role in stress, and particularly glucocorticoid receptor expression, which he showed in animal models, but also he had human data that goes along with what you were saying. And that made me think that about all of this differentiation data. Let me introduce just another idea along the lines of different regions of the brain. You made a comment very early on about dopamine neurons. And what I was thinking is, we don't really have enough data for these different brain regions, do we? To really, so it's as if this spectacular story will become even more sort of specific and interesting when we have more data from different brain regions and neuron types. Is that true? The way I'm thinking about this?
I think that you've exactly got it. Our stem cell models are very reductionist. I call them glutamatergic neurons. I don't call them cortical neurons. A lot of other people do, but I think it's really challenging to match them with a region. I think they seem to release glutamate. They could be anywhere, and everywhere. Um, as our models get better, some of the organoid models are better at patterning. I think we're going to see, terrifyingly, probably really separate results again. Like what I'm showing you is really scary, right? Because we've been doing a lot of these like RNA-seq analysis of the brain, putting them on GWAS for 5 years. And what I'm suggesting is it's not just enough to look at baseline RNA-seq, but that every possible stress or exposure that we see in our lifetime, whether it be a hormone or lead or air pollution or infection, we have to model the impact of each of these separately across all of our genetic risk, right? Like this is getting to the essence of precision medicine, but is also like not reductionist and not simple. In terms of your question about brain regions, one, I do hope that we get better at modeling that in vitro, but fortunately, Matt Giganti had the wisdom of collecting all of those RNA-seq across 4 brain regions. And so in that preprint with stress, Karina did look at trauma EQTLs across 4 different brain regions. I don't usually talk about this data because from my perspective, it really gets into the weeds. But I do believe that she had an analysis showing that the trauma specific or the trauma responsive EQTLs varied more by brain region than the base EQTLs did. So it's kind of getting at exactly what you say, that some brain region, all brain regions are more alike at baseline, and then brain regions are more dissimilar under trauma, which is unfortunate, I think, but it makes sense biologically.
Yes, precisely. Cool, thank you.
Go for it, Sally.
Hi, that was so amazing, Kristen. It's just such a beautiful example of how we can study human genetics using these cells and all of the interacting factors. So thank you for that. There were several cases where you pointed out that mouse is not a good model. I love that you're taking this to testing drugs. So my question is, what does a drug development pathway look like now? What are you envisioning to actually be able to get into the clinic?
I know this is such a cool question, right? So and I'm not saying that mice are bad models. I think they're really useful for what they're good at. But if every human is different, I don't know how we reduce them into rodents, unfortunately. Like they're too conveniently inbred, right? Like I think their strength is their similarity to each other and their weakness. And so, the real question is, do we feel good about skipping animal trials going to and so my husband's in biotech and it's something they struggle with all the time. If you find something in humans, I think it's important for safety to make sure you don't kill the rodents or the dogs. And absolutely there's going to have to be some safety stuff there to get the kinetics of how the drug ramps up and crosses the barrier. But should we be killing drugs that don't work in our terrible animal models? Like, I don't know. On flip side, when do you move forward? If you go into a trial only expecting your drug to work in 10% of cases, would you even move forward? Like, what's your ability to predict the people who ought to be responding? And I think that the obvious drugs that are going into clinic right now or into trials are those drugs that we think are going to work on everybody. We're not yet slicing that pie, but I think that the vast majority of drug discoverable space won't work on everybody. And then how do you fund those trials? How do you fund those drugs? How do we do that science? And I think that's a mess, especially this month. But it's a huge challenge. How do we cut this pie up in a way that is sustainable for moving forward?
Yeah, I mean, the good thing is that you do have efficacy in human relevant models. So, how do you sort of maximize that and use that evidence towards an IND in the future? And, your lab may not take it that way, but I think we do need to be thinking about these things. And thank you. So.
No, thank you. It's a great question. I wish the answer was easy. How?
Great. I'm just going back to the initial plot with lots of function mutations and the GWAS and I feel in genetics we have traditionally just separated these two groups, but probably the reality is that they are more connected than anything else. And have you thought about using polygenic risk scores together with loss of function mutations and use it for explaining maybe variable expressivity or penetrance or?
Yeah, so I'm so lucky to get to work with some really amazing geneticists, included amongst them Laura Huckins and Paul O'Reilly, who think deeply about polygenic risk scores all the time. They're challenging to work with, right? I mean, right now they really, really focus on this like purely additive model. And in my mind, if you inherit 20 risk factors and I inherit 20 risk factors, it makes sense to me that it ought to be different whether all 20 of yours hit neurons and half of mine hit neurons and half hit glia, right? Or if all 20 of yours hit synaptic, but half of mine hit synaptic and half hit regulatory. Like, I think what our data is showing is that you can saturate a pathway, right? And that maybe we should be considering that. The flip side being, when we tried to go back into like the electronic health records or the UK Biobank to find this data, we couldn't find these people. And I think in truth, that's because we can do extreme perturbations in cells that don't yield viable people. And so we couldn't find people with like 2 standard deviations to the extreme of like even 4 of the genes we were looking at. And so I think we can do a lot with modeling PRS in a dish, but if this is what I can model and this is who makes a viable human, it might be that why we have that disconnect between population studies that study that sliver of humans and our studies that study everything that we can do in vitro. And I don't fully know how to resolve that, but I do think it's such a cool question that we urgently need to solve, is how to do better by polygenic risk.
Yeah.
Roz, please go ahead.
Hey, Kristen. Thank you so much for giving an amazing talk. Just wanted to say that your work is a truly motivational and inspirational for a number of us who are trying to contribute to the field. So thank you so much for sharing some of your insights. I was really interested when you're talking about gene-environment interactions and when analyzing those interactions. Could you talk a little bit more about the importance of the time at which exposure to these environmental risk factors occurs and how that might impact downstream analysis?
Yeah, this is such a good question. In a world of infinite money, we'd have the time course for all of these exposures. One of the things I didn't talk about is, a big mystery to us with this H-court data is whether the PTSD derived neurons were hyper responsive or failing to recover, right? Because they would look the same. And so in the perfect world, across 50 donors, you'd have 5 time points. We don't have that data. I don't know that we'll have it anytime soon. It's a lot to ask. But it's super, like, you're right at the crux of it. And we're not looking at things like how the liver isn't, like our neurons don't exist in a vacuum of just neurons. Like the exposures they have are being filtered through all these other cell types that all also have different variants that are active in different cell types and different responsiveness to the environment. So that full computational model is nowhere close to being achieved.
Anything else?
I also want to say, feel free to
Pause.
email and follow up if you have questions, especially if they're technical. I promise you the postdocs and grad students in my lab will have way better answers than I do. And so we love working with other groups. And if anything that we did could be useful to your team, just shoot me an email and we'll connect the trainees.
Paul, did you have another question?
Well, I was just about to ask, what about somatic mutations? Have you considered that in any of the studies, or do you think they are important?
This is the nightmare, right? That a mutation in the brain that I didn't capture in the skin cell is causal. And so, in much the same way that I didn't want to think about having to test every possible environment, individually. I also don't want to have to think about testing this across the brain, but I do have a really brilliant postdoc who this is all that she wants to do is test somatic mosaicism. And so she's been working with Chris Walsh's group for the last couple of years, and she put in an MPRA all the rare mutations they found in their schizophrenia and autism cohorts and has been testing them for functionality. She found a few hits, but now we have to go back and be like, well, what percentage of a mosaic is biologically relevant? And I actually think even if she's excited and the biology is not there, we're going to learn something, right? Because if 1% or 10% or 50% of a mosaic is silent, like that's bad for mosaicism, but it's actually really good for gene therapy because it means we don't have to fix every cell. Or reciprocally, if even a 1% somatic mosaicism is damaging, that's super bad for gene therapy because it means we can't miss anybody. And so the biology is probably somewhere in the middle because it's always somewhere in the middle. But I think it's going to be really important for us to understand how to titrate what is the tolerance of the system for a couple of aberrant cells.
Great, thanks.
But wouldn't that be terrible if like you could have mosaicisms and they were all stress and environmental dependent across every, like, yeah, it gets out of control really fast.
You have to know the lineage.
Yeah, the tree of
Yeah,
mutations for the whole body.
precisely.
Yeah, for
Because
every
it's not
cell.
the risk isn't going to be equally spread throughout all different neurons. I mean, that's sort of the big conclusion of all of these data that show de novo
Yeah.
mutation causes disease. Right. So
So you'd have to catch
that's slightly
mosaicism.
mind-blowing, but
Yeah,
Yeah.
if we thought it was complicated enough, right?
yeah, right.
Every time we think we have a handle on it, the biological questions get more interesting. This has been a really fun discussion for me. These were all really fun questions to think about.
I'm glad you could come interact. This is great. Hopefully it'll be part of this sort of I think we're gaining
Oh,
momentum and gaining
yeah, no, I want to
and
double check that
interaction.
my calendar has your guys' next meeting on so I can jump in and
Wonderful.
and sneak in whether I'm invited or not.
Absolutely.
Thank you all
Thanks
so
so much,
much. Thanks
Kristen.
for setting
Yeah.
this up.
And thanks everybody for showing up. We'll see you in a month. I do try to keep the calendar up to date. If you do, I notice when some people join at odd times, if you do see something that doesn't match up or there was something on your schedule that didn't match up with what happened, let me know because I do try to keep it all in line, but I'm not so sure I always know what Google Calendar is doing. So
Nobody knows.
Thanks again, Kristen, and thanks to everybody. Wonderful to be interacting. See you soon.